Nā kutaroro, nā tororo ko ora ai te iwi. With my basket and with your basket, the people will prosper. Ana mana, ana reo, e rā rangatira mā, tēnā koutou, tēnā koutou, tēnā koutou katoa. Kia ora colleagues, Vice-Chancellor, Deputy and Pro-Vice-Chancellors, Deans, fellow professors from the University of Otago and members and guests. And most importantly, a special welcome tonight to friends and whānau of Professor Guilford, the recipient of our 2017 Distinguished Research Medal. Particular welcome to Parry's wife Annette and children Guy, Nick, Ben and Bridget, and to Annette's mother Jill and partner Glenn. Welcome, welcome, welcome to you all. Welcome also to Parry's brother Grant, who I saw somewhere before. Oh, he's up there with the whānau now, who has taken time out from a busy role at one of our other fine New Zealand universities to be here. Welcome, Grant. I'd also like to acknowledge visitors Isaiah and Kelly Piho, members of the whānau we may hear about tonight who have provided generous and positive influence and who have been positively influenced themselves by the research that we will hear about. Also acknowledge Karen Parinatai, one of our proud winners of our Prime Minister's Supreme Award for Tertiary Teaching Excellence, who is one of Parry's strong research collaborators, together with other collaborators, colleagues, friends and supporters. Welcome to you all. My name is Richard Blakey. I'm the Deputy Vice-Chancellor for Research and Enterprise here at the University of Otago, and it's my great pleasure to introduce this event to the celebration of our Distinguished Research Medal and to join you in engaging in a public lecture on the topic related to the work that led to Parry's successful application for this award. As a celebration of the occasion, we'll also invite you to join us afterwards upstairs for refreshments and conversation. We have Professor Guilford's Head of Department, Professor Kathryn Day, here to give a formal introduction for Parry in his lecture, outlining the details of his career and achievements that have led to this award. But I just wanted to give you some context as the person who has the responsibility to chair the panel that makes the recommendation to the Vice-Chancellor for the award of our Distinguished Research Medal annually. The Distinguished Research Medal is governed by regulations that state that, quote, the University of Otago Distinguished Research Medal Award aims to give recognition to outstanding performance of individual researchers. A medal commemorating the award will be presented to each recipient at a public lecture given by the recipient. So that's why we're here tonight. And there is a medal somewhere, I believe. The basis for selection will be outstanding scholarly achievement, including the discovery and dissemination of new knowledge, the development of innovative technology, or the development of concepts that lead to significant advancement in the activities of the university. And what we'll hear tonight is the kinds of work that fulfill all of those criteria and then some. Now, all of our academic departments within our 4 academic divisions are invited annually to submit portfolios from their best and brightest researcher or research team and solicit referees' reports from preeminent international scholars to support applications. Then a group chaired by myself and composed of former distinguished research medalists has the stimulating, challenging and unenviable task of selecting the best from these nominations. And it is a hard job, but it's a very special job. And what makes it so special is that we are selecting people into a highly exclusive club in this process. Unlike other awards, the Nobel Prize, for example, where there might be up to 3 awards made annually across each discipline, this is one award across all of our disciplines at the university. We have to select across disciplines, even including mathematics, for which there is no Nobel Prize. And we have to balance overall career-long scholarly achievement with the discrete, world-changing discoveries that are sometimes sufficient for other awards. And putting the scale in context, we have approximately 1,600 academic staff here at the university, with only one Distinguished Research Medal awarded annually, very rarely a joint award. And that means that the club is very exclusive. There are, 15 or 16 DRM medalists currently on staff. So that represents 1% of our academic staff who are in this very exclusive club. Consider also that to be appointed into an academic position at the University of Otago is also highly selective. Typically, we get 100 or even more application for any one position, which is another 1% selection. So already that's 1% of 1%. And then even getting to the level of a PhD in an advanced research qualification that makes you eligible for application into an academic position is about another 1% selection. Although here in Dunedin, 2% of our population is PhD qualified. We are officially the smartest city in New Zealand. And another 1% of our population is studying towards a PhD. So with those three highly selective processes, it is clear that this event that we are here to celebrate tonight is to celebrate someone who is the 1% of the 1% of the 1% in Dunedin's smartest city. So Parry, I think you are now officially the smartest person in New Zealand's smartest city, and I trust that you'll be able to dine out on that fact well until next year's award at least. But I know also you won't let it go to your head. So ati ake ne, that's enough said from me. So with that, I'll now ask Professor Day to come forward to introduce you in the topic of your lecture tonight. Tēnā koutou, tēnā koutou, tēnā koutou katoa. Kia ora.
So, tēnā koutou, tēnā koutou, tēnā koutou katoa, ko Kathryn Day aho, ko aho ti tumuaki, o ti tari matu ko ora. So welcome, I'm Kathryn Day, and I'm the head of the Biochemistry department, and I'm really honoured to have this opportunity to celebrate the research success of Professor Parry Guilford. He's a long-standing member of our department, and we're all very proud to be here tonight. So Parry has too many achievements for me to review in great detail, and you've come tonight to hear Parry and not me, so I will try and be brief. So just really to touch on a few key things, Parry had began his formal scientific training at the University of Otago, receiving a BSc and an MSc in biochemistry in the early 1980s. This was followed by several stints as a scientist at DSIR, and I'm sure Parry will mention that in his talk, and then a PhD at Cambridge in 1989 and a postdoctoral fellowship at the Pasteur Institute. He returned to the Biochemistry department in 1996 and has been affiliated with the department since then, so a very long association. And over this time, Parry has had many achievements, and really I highlighted them in three brackets. He's made seminal contributions that have been published in the very best scholarly journals, and I note his discovery of the ECAD mutation was published in Nature in 1998, and it has now been cited over 1,000 times, so that's quite an achievement. He's also done research that addresses health issues in the community and that is of benefit to the communities. And I think, most significantly, he's made a large contribution to a Maori Bay of Plenty family, and again, I'm sure he will speak of that tonight. And the third arm of his research is translation of his research into commercial products, and this, again, that is of what will be of wider benefit to the community. And again, he has been an inventor on 14 filed patents, 2 of which are granted. So these are three significant things, and I think many academics would be proud to count any one of those achievements amongst their career. And to have succeeded in all three domains is really why we're here tonight to celebrate Parry's award of the Distinguished Researcher Medal. So it's a very fine achievement. So just to sort of highlight a few other things, so not surprising, Parry has received many other awards for his research. In 2013, he received the HRC Bevan Medal for Translational Health Research. And he also that year received New Zealand Innovators Award, Supreme Winner for the development of the CX bladder test for Pacific, as in his role with Pacific Edge Limited. And in 2014, Parry was awarded the Royal Society's Sir Charles Hercus Medal for excellence in molecular, cellular, biomedical and clinical sciences. So all fine achievements. And last year, he was elected a Fellow of the Royal Society of New Zealand in recognition of these many sustained outstanding achievements. So I won't dwell further on his specific achievements, and he is far more qualified to talk to you about these tonight. However, I do want to highlight the many other ways that Parry brings great mana to the university and makes enormous contribution to the science system in New Zealand. And most notably, Parry is a member of the HRC, so the Health Research Council of New Zealand's board. He's also a member of the Cancer Trials New Zealand Advisory Board, a member of the MB Science Board, and he also has a role as a Deputy Director of the Healthy Lives Science Challenge. Again, these are 4 large roles that any one person would think was quite a task. Having all those 4 roles is really a recognition of Parry's high peer esteem and the esteem in which his colleagues hold him. So just in terms of the department, I think these accolades that Parry has received and his recognition are attributable to Parry, but have been built off of many years of cancer research in the biochemistry department. And I think it highlights the importance of taking a very long view and making strategic investments in research programs. And I think this is particularly investing in people, again, and investing in equipment that enables research. And I think it's very important that we continue to take such a long view, and that's how people of Parry's ilk can succeed. So while now for funding for Parry is very good, because this year he received an award of an HRC program grant, so $5 million over 5 years is a large amount of money, which is excellent. He also has substantial funding from the Healthy Lives Science Challenge, and this enables his research. But there have been times where I think funding has not been so plentiful, and I think again, we should remember that this great success is built off sustained strategic and thought to enable the activities and that people can flourish and prosper in their areas. And I think we're very proud to be here tonight to celebrate Parry's success. And just really, I think, typical of Parry, just this morning I had met with him and he was discussing plans for implementation of his latest research findings in the clinic, and this highlights always how Parry is thinking to move things forward. So in closing, Professor Parry Guilford has, I think, displayed a sustained record of scholarship, research excellence, and research translation for the immediate benefit of cancer sufferers or those who are at risk of cancer. It makes him a unique member of the University of Otago and an outstanding awardee of the 2017 Distinguished Research Medal. Parry, we are proud of you, and we look forward to hearing from you tonight. Thank you.
E raku ro rangatira mā. Neo ka tu ohu, I mua I a kotu, I tēnei pō. Ko e raki, toku manga. Ko Waitaki, toku awa. Ko Nadya Guilford, te iwi. Ko Parry Guilford, toku ingoa. Tēnā koutou, tēnā tātou katoa. So thank you very much, Kathryn and Richard, for those kind, if not slightly generous words about some of my achievements, which again, I must stress, are built on a teamwork and on a history of research done at Otago University and other places as well by my colleagues. And so I'm really just an example of what can be done at this university and one of many in a large team. The research I want to talk about today, I'll introduce it by a discussion on cancer and how cancer develops. So the basis of cancer, and hopefully that'll give you sufficient introduction to kind of understand the principles of cancer. Then I'll go on to talk about 4 of the areas where we've been working in the last several years to try and improve outcomes for patients with cancer. Right. So animals try very, very hard to avoid cancer. And they do this in numerous ways. They will use extremely advanced mechanisms of immune surveillance. They will use various cellular tools to repair damaged DNA. They have numerous sensors inside the cells which can recognize stress and damage and then stop the cell from dividing. They have enzymes in the cells which can detect chemicals which are damaging and which can be inactivated through chemical means. They have pumps which can pump out compounds or drugs from the cell which may cause damage to those cells. And they have a really extraordinary system of cell hierarchy and programmed cell death, which I'll explain a little bit more in this next slide. Within every tissue in the body, so by tissue I mean it may be liver tissue, it may be lung tissue, it may be stomach tissue, it may be colon tissue. The example I've got on the bottom are some colon cells. In those tissues, about 5% of the total cell content are what are called tissue stem cells. And these stem cells are immortal. They will live through the life of the animal or the organism. And they will divide at a fairly slow rate, and they'll lead to a population of progenitor cells, which will then divide rapidly to give you some other cells, which will be differentiated, and they will perform the various functions required for a cell's role in the body. So the tissue stem cells themselves are immortal, as I said, but all those other cells, the other remaining 95%, are programmed to die. So they will perform their function, then they will be sloughed off, and they will die. Which means most of the time, if there's any damage to a cell, that cell won't care because it is going to die anyway. It's going to mature for a few days. It'll carry out a job. It may make an enzyme. It may make a protein. It may make some kind of compound which is important to the cell. But then it will die, and it will always die. Which means the cell is protected dramatically, 20-fold protection from cancer risk, because most cells are going to die anyway. So if they undergo mutations or various problems which could lead to cancer, those cells will simply not survive. They will carry on and die. But the stem cells, they are the ones where there is risk. So what could possibly go wrong? We've got all these amazing mechanisms to prevent cancer. What could possibly go wrong? Well, clearly quite a lot, because we see cancer in every organism. Anything which has more than two cells rubbed together can develop cancer and will develop cancer. We see top right, we see melanoma in a snake. There's melanoma in a fish beside it. There's a brain cancer in a fly. Remarkably, you may have to believe me on this one, remarkably, shellfish get cancer. This thing here is a cancer on a coral. To the left of that is a cancer on a roundworm. Bottom row, you can see the shark. You often get told that sharks don't get cancer, therefore you should buy shark cartilage as some health treatment. Don't buy shark cartilage because sharks do get cancer. Dinosaurs got cancer. There's a bone from a dinosaur from how many millions of years ago. Also, cancer was around in those times. And amongst humans, there are 15 million new cases of cancer per year. And in the human history, it goes a long way back as well. 250 BC, one of the pharaohs was found to have died of prostate cancer. 700 BC, a Siberian or Scythian king also was shown to have died of prostate cancer. So cancer has been in the human history from the beginning of time. It is not a modern disease, it's a fundamental disease which occurs in every organism with more than one cell. It could be worse, of course. If you were a bacteria, this is an amazing fact, there's 100,000 million infections of bacteria every second. And so really, it's not so bad being a human after all. So why is cancer so common? We have all these amazing techniques the cell will use to minimize the risk of cancer. So why is it still so common? The answer is 37 trillion, because there are 37 trillion cells in the average human body. And each of these cells carries the instructions for the entire human capability. That is 20,000 genes, all the genes which encode every function that there is in a human body. So if you carry those 20,000 genes, you also carry the manual for cancer. So fortunately, differentiated cells, and 95% of cells, have critical instructions redacted from their DNA, their chromosomes. And this is done through epigenetics, where the genes will be silenced by turning the DNA so it tightens and the gene cannot be turned on. So most genes are turned off. But that still leaves 2 trillion cells, which are the stem cells, which really have a fairly clean copy of the body's genetic instructions. So 2 trillion cells have access to 20,000 genes and all the capability that 20,000 genes can bring. So cancer begins in tissue stem cells, and it begins through the activation of capabilities which are normally used by specialist cells. So for example, they can proliferate rapidly, just like cells in a healing wound will proliferate. They can migrate like cells in the embryo will migrate. They can move in and out of blood vessels like white blood cells do. They can coax blood vessels to grow into them to provide oxygen, just as you would see in repairing tissue. They decrease their reliance on oxygen, just like oxygen-starved tissues do. And they can hide from the immune system, just like cells in the colon often do. So all these tricks that are used by a cancer cell as it develops and gains its capability are really just taken from the normal repertoire of skills that a normal body has. But because the cancer has started in the stem cell, it has access to all the instructions, the 20,000 genes, and it uses those to acquire the characteristics it needs to be a fully fledged cancer. So of course, all this means that drugs that kill cancer cells will also damage normal cells as well. We cannot get around that fact with any ease at all, because the processes of a cancer are the same as processes of the normal cell. You will always find the drugs which you can use to treat cancer will cause some damage, some side effects in the normal body as well. It's not like a virus, it's not like a bacteria or a fungus, which brings in foreign techniques, foreign biochemistry. These are processes which are normal to the human body, and therefore you target them in a cancer, you'll also cause damage to the normal tissue, which is really the main reason why cancer has been so hard to treat over all these years. So cancer has acquired all these amazing capabilities, but how does it actually do that? We know it can see the genes, it has those genes there, but how does it actually acquire the right ones to become, to grow, to invade, to become metastatic? How does it actually achieve that? Well, there's only 2 steps required to do that. The first one is the need to acquire DNA damage. So the genes, the 20,000 genes, must undergo some kind of damage which changes their activity. They'll either be turned on or turned off. And so the cancer will be able to acquire new capability by genes being turned on or turned off. So DNA damage can occur in many ways. It can be caused by the environment. So for example, carcinogens in the environment. So bad nuns get lung cancer. I don't know whether good nuns get lung cancer or not, but I suspect they probably do as well. You also, of course, have radiation from the environment can cause DNA damage, another example of environmental factors. You can also get epigenetic changes caused by the environment. So this is when the genome is effectively tightened or loosened to enable access of various factors to the genes which are involved in cellular processes. The example I've shown here is of a little boy from the Dutch hunger winter. So in 1944 and 1945, the last 2 years of the war in Europe, there was a blockade by the German troops around the urban areas in the Netherlands. And this lasted for almost a year, and about 4.5 million people were blockaded off. 20,000 people died during this blockade, and there was intense starvation in this area during that period. But since then, researchers in the Netherlands have followed many of the people, those 4.5 million people, to see how they have developed, how their lives have changed or been affected by that period. And what they observed, amongst other things, was that the people who were very little children or were still in utero at that time have grown older, but with a much lower rate of colon cancer. So what this means, we're not too sure, but you could perhaps suggest that maybe one reason why we have such a high rate of colon cancer in New Zealand is the opposite of that. We have a nutritional excess rather than a deficit. But that, of course, is speculation. So the last and probably the most important single cause for DNA damage is bad luck. There are 3.2 billion nucleotides in the human genome, and you get 1 to 3 random errors or mutations occurring every time a cell divides. And so this causes the changes to DNA, which can be occurring in important genes involved in the processes which lead to cancer. So step 2, so this is how a cancer acquires this capability. Step 1 was DNA damage, which provides a diversity in the genetic material. Step 2 is simple natural selection, just as as Darwin saw with the finches in the Galapagos Islands, just as we see as humans have evolved from apes through to what we are now, just as we've seen snakes evolve into lizards. It's the same principle here, natural selection, where there's been a selection for cells with a growth advantage. And surely that's all that cancer is cells with a growth advantage. This impact of mutations isn't just causing the evolution or the initiation of cancer. It's also very important for how cancer evolves and develops even through the course of treatment. So there's a few numbers here which I won't go through, although I know half of you will be kind of tearing through them right now because, being scientists, you probably will, but don't worry about them too much. But the basic idea of those numbers is that inside a cubic centimetre of tissue, there are 5 by 10 to the 8 cells. So a remarkable number of cells in a cubic centimetre of tissue. And so if that is cancer tissue, there's a lot of cells in there which undergo mutations all the time, which means every nucleotide in a tumour of that size could be mutated per year somewhere in that cancer. So some cell in that tumour could be mutated, every nucleotide in that in the genome could be mutated somewhere in one cell in that tumour, which means that the mutation that you don't want to see because it's going to give you resistance to your drug is already present in the starting tumour, which means, of course, that the treatments that we give patients now will always lead to, almost always lead to outgrowth of a resistant clone. You will see a strong response early on where the tumour will shrink and die away, but then there will be cells remaining which have a mutation in them which gives them resistance, and they will grow out in time. That is inevitable, and it's something which we've struggled to accept, but it is inevitable. This has been shown quite nicely by a study by Charlie Swanton's group in the UK a few years ago, where he's taken a primary kidney cancer, so that's a kidney tumour there, and he's taken different samples from different points in that kidney tumour, and also the tumour is also spread around that patient's body. So I think there's lung mets, there's chest wall mets, so there's multiple sites, and he's biopsied multiple sites of this cancer, and then he's done DNA sequencing to identify the mutations which have occurred in this kind of whole tumour burden. And what he saw was this kind of evolution of mutations, and he could build these trees, these phylogenetic trees, of showing how the mutations have evolved in different subclones, different branches of the tumour mass. And what's striking about this is not so much that observation, but for me, it's the similarity to Darwin's tree of life. I sometimes call it his I think picture, where he first described this notion of evolution and the evolution of the species. So exactly the same idea. We see evolution occurring in a really rapid time frame in a tumour in a patient. So evolution has been remarkable for providing humans with their complexity and their performance, but it has come at a price, and that price is cancer. So cancer is inevitable, unfortunately. The wheels will fall off at some stage. But a lot of people don't get a fair crack at life. A lot of people will not be around to see their niece get married, to see their daughter graduate, to see their grandchildren born, to travel, to do whatever they want to do. They miss out on those opportunities. They may not even get a chance to force a swan into the back of an old car. They will miss the full richness of life. And so, therefore, we must continue with cancer research. Even though it is inevitable, we must continue with cancer research because not everyone gets a fair crack at life. So now I want to go and move into telling you some of the work we've been involved in. So 4 short stories of the research that I've been involved in over the last few years while I've been at Otago University. These 4 stories all have common features. They're all translational, which really just means we've always had the patient or clinical outcomes in our focus. It has sometimes and does still involve basic research, but always the focus, the end game has always been how do we get this latest research to be applied to patients, to the clinic as soon as possible. And so even though you may be doing basic research, sometimes there'll be a crossroad and you'll think, well, I can go that way, which will be really interesting, or I can go that way, which will be a faster route to improve patient outcomes. And so the translational mindset will send you down that faster route each single time. But I should also stress all the translational work is built on a basis of basic research. We are relying on years and years of basic research done by people who have come before us. So we cannot be translational without the work of the basic scientists. The research in these 4 stories is all highly connected in that very often it involves families, very often it involves work with patients and patient samples, very often it involves work with the clinical teams, the oncologists. It's also very implementation heavy, which means vast amount of work has to go in. You know, once you've made your initial discovery, you've got your initial idea, a vast amount of work has to be done to actually make that new idea something which can be used, something which is robust and can be used in the clinic. And it's people like Tumi Torosin up there who's done the hard yards and a lot of this work as well. And that is remarkably important. It doesn't get the same level of publications, but it is the critical part of translational research is implementation. And I applaud people like Tumi who do that so well. Also, each of these stories shows incremental improvement, and each is imperfect. And I stress this because I think sometimes we're afraid to try new things because we're afraid of failure. We're also afraid to not do something perfectly. But it's actually often we need to just make that small incremental step. And I think this has been observed clearly by many others before me. In particular, Robert Watson Watt, the inventor of the early radar warning in World War II. So Robert Watson Watt said, give them the third best to go on with, the second best comes too late, and the best never comes. And Churchill, the guy, the one with the moustache, said, the maxim nothing prevails, but perfection may be spelled P-A-R-A-L-Y-S-I-S. Okay, so I think it denotes the same idea I'm trying to get across to you, that you really have to try things, even though your ambition you may not get to the perfect goal, the perfect diagnostic test, the perfect treatment, you can still and you must still try and strive towards that goal with something perhaps imperfect. When I was looking for this Churchill quote, I found another one by Churchill, which is unrelated, but I did find kind of amusing. I only believe in statistics that I doctored myself. And I heard Mick Black using that same line just this morning. Right, so now into those 4 stories. The first story goes back in some ways to 1864 and the Battle of Gate Pa. So this is the battle between Ngāi Tarangi in Turanga against the British troops, and it was a famous battle because the iwi were surrounded by the British troops, who just decided to bombard them with missiles for several days until, of course, they all would capitulate and they could walk into the Pa and just take over and have a glorious victory. And so they did that. They bombarded the Pa for several days from several different angles, and then eventually the whole thing was reduced to rubble, they thought. So they okay, now we will march in. They marched in and found that the Maori had dug trenches and various hideaways and quite a maze in there. And many of them were just fine, thank you very much. And so the British got a fair thumping, and it was a very famous and very, very important battle. For 2 reasons. One was important because it is said that if the British had acknowledged that defeat, they would not have got involved in trench warfare in World War I the way they did. But also it was important and famous for the humanity shown by the people of Ngāi Tarangi, because when the battle was raging, people from the iwi, in particular women, not so much this guy here, but particular women, were sneaking into the Pa site to bring water to the wounded British troops. And so the leader of the British troops, Lieutenant Colonel Gamble, said, Ngāi Tarangi are an enemy, anything but despicable, either in intelligence or courage. So that was in 1864. I am getting there towards something to do with cancer. So 100 years later, 1964, we see a paper published in the New Zealand Medical Journal by a guy called Ted Jones, who some of you might recall. He was a graduate from Otago University as well, a medical graduate. And he described in the journal a big family tree, in which at the top there in red is a princess of the Ngāi is married a Pākehā male. And from that marriage, we see a subship of numerous children who have gone on to develop stomach cancer. Ted also said the present survey on stomach cancer seems to have done little more than imbue the family with a deep sense of makutu or curse. The curse that the family believed had befallen them was due to the land behind the Wharanui here, so just up to on the left, on your right-hand side, you see the remains of a quarry there. And that land was quarried in the 60s by the city council to get rocks to help build the port in Turanga and the railway line into Turanga. And of course, the people have been in this site for 800 years. And so when the land was dug up, there were bones found. The land was in fact sacred. It was tapu land. And the whānau had believed that they were cursed because they had sold the land to the city council, and therefore they deserved the cancer in their family. The truth is, the land was taken from them under the Public Works Act, so they never deserved to be cursed in the first place. So, to 1995, Ngāi again, there were a group of 4 of them in particular who showed the intelligence and the courage to say, well, this is not caused by a curse, this is caused by genetics. And they went out and found initially Tony, Gil, and eventually myself to help understand how the cancer was occurring in these families and how whether we could develop a test to identify who was at greatest risk in these families. And that led through numerous years and numerous many hours of work to us describing a new cancer syndrome called hereditary diffuse gastric cancer, or HDGC, which is caused by dominant inheritance of germline CDH1 mutations, so a gene called CDH1 or ecadherin. And so people who inherit a germline mutation in the CDH1 gene or ecadherin gene have a lifetime risk of about 70% of getting diffuse type stomach cancer, or diffuse type gastric cancer. Same thing, I swap between gastric and stomach, it's the same condition. And they also have, the women have about a 40% risk of lobular breast cancer as well. So since we found that first mutation in what we call family A in Ngāi, there are at least 500 families worldwide have been identified with germline CDH1 mutations. And so what first started with the efforts of a whānau in Tauranga has now led to a clear cancer syndrome which has benefited hundreds and hundreds of families around the world and thousands of individuals who carry the germline mutation. What is quite striking in New Zealand is that there's around about 20, probably a few more now, families in New Zealand, of whom 13 have of Maori origin, and probably about 400 Maori carry mutations in the CDH1 gene. One of our students in the lab, Chris Hackard, recently showed that about 13% of all Maori diffuse type stomach cancer cases have a germline CDH1 mutation. So these figures are probably a lot higher than the rate you would see in non-Maori, in Pākehā families, and it suggests perhaps that Maori may have higher instances of this mutation because of some evolutionary advantage, and I'll touch on that point a little bit later on. So we're able to, with our international colleagues, build up some guidelines on how best to manage families with germline CDH1 mutations. So the basic idea is that at about age 16, you'll have a gene test, so a blood sample is taken and they'll search for the mutation in your family. And then the recommendation, if you have the mutation, is that you'll have a gastrectomy, you'll have your stomach removed, just the whole thing removed, and then just rejoined. A little bit more complex than that, but that's the basic idea. And if you don't want to have your stomach removed just like that based on the gene test, you can undergo surveillance by endoscopy every year or so, and then if they find a cancer, then you will have your stomach removed. And that process of surveillance will carry on until such time as something is found, or perhaps you might go many, many years and be okay. So that is the way it's dealt with. So has it been useful? Well, yes, it has. This is the Ngāi Tarangi data. I think it's the only graph I'm going to show you, fortunately. So this is the cases in that iwi. So they had 5 to 7 cases every 10 years for probably as far back as we can remember. And in the last 5 years, there's been no cases, and so clearly the cancer is under control in family A in Ngāi Tarangi. And it's the same story in families all around the world. This is an amazing family from Wisconsin. So in the middle is Karen Shelcom-Scriber. She is the founder of a HTGC support group called No Stomach for Cancer. And here she is with her brother and her nephew showing off, we would call them scars, but she calls them their lifelines. Those are their lifelines. And so amazing family, and they're all doing extremely well because they've chosen to have gastrectomies, and therefore they have no risk of this disease. In this photograph, this is taken in Philadelphia at a meeting organised by Karen for families with HTGC, and the only galoot in that photograph with the stomach is the one lying down. Everyone else has had their stomachs removed. And you can see most of them looking really well. 1 or 2 are still quite soon out after surgery, still not looking so well, but most of them are put on good weight and they're living good, healthy, practically normal lives because of the surgery. So as one of the kamatua in Ngāi Tarangi said to me, perhaps things are getting better now because the hill is beginning to heal. Right, story number 2. Tumi Tora up there in the photograph. This is about Pacific Edge. So in 2001, Tony Reeve and myself and a few others founded Pacific Edge Limited. And we founded it because we were frustrated by funding. We could keep on going with our research, but we never had enough money to implement our research. So we could get the basic ideas in place, but we never had enough to take it forward and actually change clinical practice. So Tony and I were determined to find new sources of money to bring that about. And so we went down the commercial route to try and achieve that. The underlying technology was what was called Microarray, and simply it was a way to look at the expression or the activity of every single gene in the genome in a single experiment. And up till then, we'd done things a gene at a time, 1 gene out of 20,000 at a time. You come up with a hypothesis, you go, I've got a really good idea, and you would do an experiment and you go, oh, well, that wasn't right. I've got another hypothesis. You'd repeat the process until eventually you would make some progress. But with Microarray, you could look at every single gene, a single experiment, and it was therefore remarkably powerful because you could find changes much more easily in gene expression changes in cancer, and you could always understand the context of any particular change. And so incredible technology which was unbiased by our rather poor hypotheses. And so Pacific Edge has gone on to use that technology to develop a test for bladder cancer. And the logic of the test is really quite simple. As a cancer develops inside the bladder, cells will fall off that tumour and they'll end up in the urine. Those cells, being cancer cells, will have certain genes more active than other cancer cells. And so what the company does is it measures the expression of 4 cancer genes, and they have high expression in the cancer cells compared to the normal cells. It's as simple as that. But the real job has been to implement that technology into clinical practice, and I'll come into that more on the next slide. But what the company has done is now developed 4 different bladder cancer tests which fit different parts of the clinical needs. So, for example, there's tests for early screening, tests for people who present with symptoms, and a test for people who have been monitored for recurrent disease. So, incredible effort, but a vast amount of that work is implementation research, which is very, very hard yards. This is just a slide which just gives you some idea of how the test is working and how the test can be useful. And the test is called CX Bladder. This is one of those tests. So what's been done here is a group of 18 urologists have been given data from 30 patients who have, real patients who have are being treated for possible bladder cancer. And so the data has been analysed by each of the clinicians, and they've decided what treatments they would do, what procedures they would do. Would they do cystoscopy, invasive or flexible? Would they do a contrast CT scan? Would they do a pilogram? And so that's all been done. They sit down, they looked at those 30 patients, made their decisions on how they would deal with those patients, and then they've been given the CX bladder, the true, actual CX bladder results from those patients. And then asked the same question again, how would that change your management? And so here, if the box is green, it means they would do less, they'll do one less procedure than they would have before. If the box is red, they'll do more procedures. And so above the black line are the patients who had a positive CX bladder test, and you see they're getting more procedures because they are positive for the CX bladder test. They almost certainly have bladder cancer, and so resources have been piled into that group of patients. Whereas the big group below are the ones who are CX bladder negative in their test, and the clinicians now say, well, it's okay, we can take resources away from those patients, they'll probably be fine, we don't need to give them nearly as much intensive treatment. So what we're seeing overall through the work in the company is a 15 to 44% overall reduction in invasive procedures. Depends on whether it's for surveillance or whether it's for monitoring, what that reduction is, but around about overall about a 30% reduction in the number of invasive procedures. And so this has now been picked up, fortunately, by the DHBs in New Zealand, and I think it'll even hit the DHB in Dunedin before too long. And it's very, very powerful technology because it's pushing resources away from those who don't need it onto those who most need that technology. Story number 3 is circulating tumor DNA. So circulating tumor DNA is DNA which is released by the tumor into the bloodstream. And you recognize circulating tumor DNA by sequencing the blood and identifying the presence or absence of mutations and genes involved in cancer. So the great strength of this technology is it is highly specific. You don't have mutations in cancer genes in general unless you have cancer. So it's a technique which you can use which is very, very specific. You don't have high backgrounds as with other techniques. And it can be used as a personalized assay to measure individual disease burden. For patients who have been diagnosed with disease, you can use it to follow their progress through treatment. We have a project funded by Healthier Lives on circulating tumor DNA. We were looking at colorectal cancer and melanoma, and we've also got other funding from other sources, including the HRC for stomach cancer, various other groups for breast cancer as well, and even, we hope, pediatric cancers. And we're asking the question, does ctDNA, circulating tumor DNA, enable us to predict treatment response faster than standard technologies such as CT scans? And then, most importantly, we have to adapt the technology to New Zealand conditions. So this is an international, we're not the only group doing this is an international initiative. But we have to adapt the techniques to the New Zealand conditions. And we also have to build some kind of implementation pathway, which I stressed earlier on is terribly important to getting things done. We have to spend a lot of time on implementation, but we'll build a pathway, hopefully with Healthier Lives funding, for all cancers. So to give you an idea of what we mean by establishing an assay for New Zealand conditions, I'll give you one example of work done by one of our very good students, Sarah Parry, where she looked at the stability of circulating tumor DNA over time. So when the sample is left on the bench, but in a particular tube, was it stable or not? And she found that using a particular tube called a Roche tube, the DNA was stable for 2 weeks. So that's great because in New Zealand, you know what happens, don't you? If you live in Karamea and you have a blood sample which has to go to a lab somewhere in Dunedin, it will go on the bus. And it'll go on the bus and it'll come down and it'll go down to Lewis Pass and it'll go down to Murchison. It'll do a little loop around there. The bus will stop in Christchurch for Easter. It'll restart. It'll go up to Mount Cook, pick up a parcel, come down over the pig route and be back in Dunedin after about 11 days, 12 days. We know our sample will be stable for 14 days. And so that's what I mean by adapting to New Zealand conditions. We have to be able to do things in a way where it's done in New Zealand style, which can be quite slow. We need to be on top of the costs because we can't simply buy something off the shelf from Roche or some other big US company because you can guarantee we won't be able to afford it all. We need to have the bonnet open. We need to understand the mechanism of these tests so that we can adapt them, keep the costs down, and use them best for the New Zealand population. So here's a bit of data from 4 patients from Dunedin who were followed through the course of their treatment. So these are all patients with colorectal cancer, and they're undergoing chemotherapy for that cancer. So the arrow shows the start of their treatment. So you see the first patient there, patient A, they've got a very high level of a particular mutation in a gene called APC. So I think it's 1,400 copies per mil of plasma. And that has fallen away in 2 weeks' time to being undetectable in that patient, and then stayed at undetectable levels throughout the course of that patient's treatment. So a fantastic result. Clearly the treatment is working very, very well in that patient. Patient B, pretty good, fairly rapid decline in the markers with a particular marker in the TP53 gene. It's declined quite nicely, but it's starting to plateau out, hasn't quite got to being undetectable, and at the end a hint of a rise in that last cycle. So perhaps already we're starting to see resistance developing in that patient. So the bulk of the tumor has been killed, but there's been some cells that have stayed behind, which have now said, now I've got room to move, I've got more blood, I've got more space, I've got more oxygen, and I'm now going to develop and grow. So maybe we're seeing that in that patient there already. Patient C, well, that's a different story again. The patient has just laughed at the treatment, or the cancer has at least. And so the tumor has continued to grow throughout the treatment. So 2 weeks, it's probably doubled in size, and even higher and higher as the treatment has gone on. So despite an intensive chemotherapy regimen, this patient's tumor is just growing through that treatment. Patient D is one where we see a different kind of pattern. So initially there seems to be a growth in the cancer at the start of treatment, but then perhaps, I haven't seen the clinical information yet, but perhaps there's been an adjustment of the treatment for that patient. Maybe they put another drug on. Maybe they've increased the dose, maybe something else has changed, but it started to fall away again, and it's starting to drop away quite nicely and held at a low level. So this is the type of data we can see, and we can do this really for practically every single cancer patient that there is undergoing intensive treatment for cancer. And so you see how it gives us a nimbleness which wasn't available before. Previously, you would have to wait 8, 10, 12 weeks to have a CT scan, and then the clinician will say, well, it looks like your cancer's getting better, or it looks like your cancer's getting worse, so we'll try something else. Now there's a chance to actually move the whole game into a matter of 1 or 2 weeks. You can say the tumor is clearly responding very well to this drug, we'll carry on, or vice versa. The tumor is just not responding to this drug, let's change it immediately. So incredibly powerful technology which applies really to most cancer patients. So the applications we think of for circulating tumor DNA are the obvious one is you can stop futile treatment. You don't keep a patient carrying on through chemotherapy for 6, 8, 12 weeks when it is doing no good at all. You could think about new ways to reduce or increase drug doses. So if the patient is doing well, so one of those patients who drop down to 0, maybe you could think about easing back those doses in a really safe, rational way. Ease those doses back so there's less toxicity, the patient feels a lot better, and you can just keep a very, very close eye on those levels. And if they start to rise, you might have to increase dose again, but it's a way to modulate the treatment that you give to patients almost in real time. And I think it'll also lead to possibly a new science of maintenance therapies where you develop new types of drugs, new ways to treat patients which will maintain a drug as a chronic disease rather than something which is more explosive than that. So that's something which I think we'll see more of in the future. And it probably will require more thinking about population genetic principles. Importantly, I think it gives the oncologist a chance to do very quick assessments of new treatments. So I think they're often, quite rightly, they're very concerned to try a treatment on the off chance it might work when they have to really give it several weeks to be confident they're giving it a fair go. With this technology, you could really try something and within 2 weeks make a decision whether the treatment is one you want to keep or change to something else. It could be used to confirm uncertain CT and PET findings. So quite often people will have a scan and there's a strange looking shadow or lump there and they go, what's that? And not quite clear what it is. There's a real chance to use this technology to actually make a much clearer diagnosis or clarification of CT and PET scan data. You could identify tumor heterogeneity. So by sequencing the blood, the ctDNA straight from the blood, identifying new mutations, you can see how a tumor might be evolving and therefore choose new treatments to treat that patient. And we've also just started a project recently where we're going to try and develop circulating tumor DNA as a primary diagnostic tool. And the initial focus will be on the Pacific Islands, where, of course, current diagnosis is probably presented with a large, obvious lump. If we can take this technology to places like the islands and get a simple blood sample sent to either a lab in New Zealand or a lab in the islands, we might have a way to get much earlier diagnosis of cancer in patients in those resource-poor areas. We could also reduce the reliance on CT PET scans, which of course are expensive, they require a hospital visit. And so what we hope we can do with this is migrate surveillance away from the hospitals, more to the community, more to the primary care setting. So this will be very important, I think, to reduce inequalities in healthcare, and that, for example, Maori Pacific people will be able to have their care in the communities on the marae, for example, and with less need to travel to the hospitals for CT or PET scans. And the same for anyone in the rural community, they will be able to have a follow-up checkup by simply providing a blood sample at the local health clinic or with the rural health nurse. So great opportunity to reduce inequalities, both in Maori Pacific people, but also in rural populations. So story number 4 is the prevention and treatment of CDH1 mutant cancers. So I told you before about HDGC, the stomach cancer syndrome, which is caused by germline mutations in CDH1. We have a real need to try and develop better treatment. So the gastrectomy, the removal of the stomach, it works. And I'm proud to say we've got people in the audience who have had that procedure, and they look fantastic. But I think they would also agree that there must be a better way than such serious surgery as a gastrectomy. And so we're working on trying to develop drugs which will prevent the development of cancer in those families. Also, those CDH1 mutations are really common in sporadic cancer as well. So not only in families, but also in people without a family history. So diffuse gastric cancer, there are 400,000 new cases per year of that type of cancer. Lobular breast cancer, 150,000 new cases a year. So really quite common cancers. They have CDH1 mutations, and therefore anything we develop for CDH1 for the families will be also very useful for a much wider population base as well. So how are we going about doing this? CDH1, or E-cadherin, is a tumor suppressor gene, which means it's a brake on a cell. And so it's inactivated in cancer. So the brake is removed in cancer so the cancer cells can divide more rapidly. And they can divide more rapidly, and they'll also migrate more rapidly. They'll move around the body more easily, more rapidly. So you get more rapid invasion, more rapid metastasis when there's an E-cadherin mutation. But this loss of E-cadherin must also come at some other cost. It can't simply be all gain. You cannot simply get, in a cell, something as evolved as a human cell, you cannot get all gain by knocking out an important gene like CDH1. There must be a cost somewhere. And so what we're trying to do is work out what that cost is, and when we find that cost, how do we exploit that cost? So just to keep it very, very brief, we've been trying to identify vulnerabilities in cells which have mutations in the CDH1 gene. And basically what we seem to see is that the membrane is disrupted in these CDH1 mutant cells, and so that they don't signal correctly inside the cell. We see various processes such as endocytosis isn't operating correctly. We find numerous receptors in the cell surface don't work correctly. And I think it all comes back to a failure of those cells with E-cadherin mutation to kind of connect correctly with the inside layers of the actin cortex. So we think that is an area of great vulnerability in the E-cadherin negative cells, which we can target with drugs. In fact, we're seeing that already. We've found using various chemical inhibitors, various drugs, we've been able to show that we can inhibit various types of receptors, various processes such as endocytosis, such as cholesterol distribution in the membrane, and see more death in the CDH1 mutant cells than in the wild type cells. So we think we've got a way in now to treat cells which have a mutation in CDH1, which will leave the CDH1 wild type or normal cells relatively unscathed. I mentioned earlier on that we see this higher rate of CDH1 mutations, germline mutations, in Maori families, and asked, does it perhaps indicate a increased or an advantage in an evolutionary sense for Maori people by having those mutations? And we did some work some years ago where we showed that certain E-cadherin mutations made those cells partially resistant to infection with the bacteria which causes listeriosis. There's also been a recent paper which talked about the migration of Maori from the Cook Islands in about 1450, and they suggested that migration was driven by an outbreak of food poisoning caused by the Ciguera toxin. So this is a toxin which is produced by an algae which accumulates in the reef fish and then eventually gets into the human population, and it causes very intense food poisoning and often cardiac events and death. And so this paper described the likelihood that that might have occurred in the mid-1400s and driven the migration from the Cook Islands to New Zealand. And so it makes you wonder, because this toxin works by activating voltage-gated ion channels, and we know in E-cadherin negative cells those channels aren't working quite right. So we would hypothesize or speculate, more correctly, that perhaps the E-cadherin mutation has given a degree of resistance to things like Ciguera toxin or Listeria or other bacteria through evolution, and therefore those mutations have been selected for. Today, of course, it's not a good thing because people live a longer life, but perhaps in pre-European times, I think the average life expectancy was about 35 years, so you probably wouldn't see much cancer then anyway. So just speculation, but I think it's worth giving some thought. So really just now to finish, just a few thoughts on how I see cancer treatment in the future and how I think cancer will be seen as a chronic disease in years to come. Just as we now see diabetes as a chronic disease, I think we'll see cancer as a chronic disease in years to come. There'll be progress on all fronts, I think. So in the area of prevention, I think we'll start to find low-frequency, high-impact environmental risk factors, which are causing cancer. So at the moment, the focus is on factors which are common across the population, like dietary factors like smoking, which are very common across the population, but have some of them have quite a modest effect. Not smoking, but others have quite a modest effect. Bacon. Does bacon really cause that much cancer? Well, it does cause a bit of cancer across the whole population. Quite a big deal, probably, but for an individual, maybe not such a big deal. But I think we're going to see in future, by using genetic methods to identify people at highest risk of certain mechanisms, we're going to find that some people with certain genotypes will have higher, very, very high risk of particular environmental risk factors. And I think we'll see a lot more work in that area in the future. I think we'll see a lot more use of chemoprevention to reduce cancer's selective advantage in high-risk groups in particular. In terms of screening and diagnostics, I think we'll see a lot more use of indwelling diagnostic devices, so things we put inside the tissue, inside blood vessels, to sample the blood in real time to give us data on the presence or absence of particular cancer markers. Probably circulating tumor DNA markers will be amongst those. And those will, of course, now allow us to find cancer early and cure the cancer by surgery. In treatment, I think we'll see a shift towards the application of population genetics more often. So you'll control the tumor in a way where you try and avoid outgrowth of more aggressive clones which you do not have treatments for. So you'll try and maintain a tumor there. You won't destroy it, you'll maintain it, but you'll control the outgrowth of clones which you know you will struggle to cure. I think there'll be increased use of immunotherapy in coming years. And importantly, I think we'll use drug combinations which anticipate resistance right at the start of treatment. So you will not wait for the resistance to develop. You'll know it will come in time, and so you will treat with a combination of drugs early on which anticipates the later resistance. And I think importantly, as I mentioned earlier on, there'll be more treatment flexibility because of the use of circulating tumor DNA. I think the doctors will be able to just try things for a short period of time, see how it goes, and mix and match things much more quickly and easily than they do now. So to me, that'll be the major changes we'll see, which I think will turn cancer into a chronic disease in how long in my lifetime. So the research carries on because it is our common cause, because we care for those amongst us who are struggling through no fault of their own, and because there we all go but for the grace of God or the throw of the dice. And with that note, I'll acknowledge a few people. Of course, my own family up there and the several rows back. Wonderful to see you there. I want to, of course, acknowledge the families, particularly the families involved in CDH1 research. We've got 3 members of one of the families just there and one of the front rows as well. Without their support, their encouragement, we would not do any of this work. Kimihaura Health Clinic, which is the one from Ngāi Tarangi, who did all that early research in CDH1. Of course, they were the drivers of that project. The Cancer Genetics Laboratory, all the people from that lab, wonderful group of people. Department of Biochemistry, a great laboratory, a great department to work in. So thank you very much, Kathryn, for creating that environment, and Kurt before that as well. Pacific Edge Limited, people like Toomey up there, and Tony Locke somewhere up there as well, and many others from the company who do all those really hard yards. We can come up with some ideas, and that's quite easy. It can only take a year or two, but it's the hard yards done by people like Toomey and Tony, which get these tests into the clinic, who really need the applause and the congratulations. And finally, of course, I must acknowledge one of the wonderful funding organisations, the Health Research Council in particular, Healthier Lives, New Zealand Breast Cancer Foundation, and the Karen and Louisa Trust. And I'll leave it there. Thank you.
Stand over there. Yeah, just stand over there for a bit, Parry. Got a few more words to say before we invite you all for refreshments, because we have a very important thing to do. We have to invite the Vice-Chancellor up to award you the Distinguished Research Medal. But I just want to comment on the significance of your lecture before that very briefly. Firstly, for you, the significance is, we didn't tell you this is a test as well. Giving the lecture is a requirement before you receive the award, and in fact, you're all examiners. And so if you think that Parry has shown us sufficient excellence, then please join me in thanking Parry again. So what I've taken from your lecture tonight, Parry, is that you exemplify everything that we want to see in our outstanding researchers, that again, in our national framework of research excellence and impact are pillars that we stand upon, and where excellence is a necessary but not necessarily sufficient condition or requirement for much of our research. If you find a way to translate research into something that will be of use to people and communities, you have an obligation, and you actually take pleasure in seeing that research translated. And we've seen that tonight with your story of evolution, families and cancer, how built on basic understanding from yourself and many, many others, you've found ways and told us in 4 stories how that research can become translational or aspire to be translational. And I know you've got a lot of other aspirations with people like Karen of actually saying that this will be a story of evolution, families, whakapapa and cancer, and I know that some of our funding agencies were not quite wise enough to allow you to explore as quickly and rapidly those aspects of incorporating whakapapa and Indigenous knowledge into your research to allow better understanding and better translation. So I'm a very strong advocate for the kinds of things that you are doing with your colleagues in expanding that realm to get between those pillars of excellence and impact ideas of collaboration and translation to get the best out of the research for the people for whom it matters, the public of New Zealand and around the world. I think we've all seen a wonderful talk. I won't say any more because I'm the one standing between you and your medal. So I'd now like to invite the Vice-Chancellor, Professor Harleen Hayne, to come and present you with the 2017 Distinguished Research Medal.
So it is just left to me to thank everyone for attending this fantastic event and to invite you all to join us for some refreshments in the College of Education tea room, which is just in the back of the auditorium. And on behalf of the University, Parry, I think you can see that the audience all agreed that this was a very well-deserved honor. I think one of the things that has always impressed me about Parry's research is that it's not only world-class, but it's also very useful. So it's a great honor to present the medal to you today. Thank you very much.